I, Dr. Sonali Mahapare, Associate Professor and Head of Department of Pharmaceutical Chemistry, Dr. D.Y. Patil College of Pharmacy. Today we discuss bioanalytical method development and validation. So, bioanalytical method development and validation according to USFDA guidelines. First, we'll see introduction, objective, method development, method validation, validation parameters, and references. So, what is bioanalytical method? Bioanalytical method used to determine the concentration of drug or its metabolite or both in biological matrix such as plasma, serum, urine, or any other biological fluid, etc. Used in human clinical pharmacology, viability, and bioequivalence study. Also, it is required for that pharmacokinetic evaluation. It is used for non-human pharmacology or toxicology, that is preclinical studies. So, bioanalytical method development according to that USFDA guidelines, which will be useful for the determined concentration of drug or its metabolites in different formulations and which is present in the biological fluid. So, bioanalytical method is used to develop and validate highly specific, reliable, accurate, precise, cost-effective HPLC, LCMS-MS, LCMS, GCMS method for determination of drug and its metabolite in human plasma or any other biological fluid. The scope of developing and validating the bioanalytical method is to suitable method which is most accurate and precise for the analyte. So this bioanalytical method which is very useful for the determination of the drug or its metabolite present in the plasma, serum, or urine with the help of that HPLC, LCMS method or GCMS method. There are different types of validation, method validation. There are three types: full validation, partial validation, cross validation. Next, steps in method development. First, with the help of literature survey, we have to find out drug, identification of analytical techniques and its optimization, reference standard preparation, selection of internal standard, sample pre-treatment, that is extraction procedure. You have to determine extraction procedure like protein precipitation method, then liquid extraction method or solid phase extraction method, and after that storage of sample, and then we can determine that method validation. So first we have to do that literature search of the drugs. First select that drug, find out formulation and its dose, find out that determine that pharmacokinetic profile, that is Cmax, Tmax, protein binding, then physical chemical properties like chemical structure, molecular formula, solubility, also pH, pK value. Then next step, that is identification of analytical techniques and its optimization. So first you have to identify that analytical technique, like we have to determine HPLC or LCMS method or LCMS method. So first we have to mode of separation, that is separation of drugs. Then if you select that HPLC method, then find out selection of that column, selection of mobile phase. So we have to select that mobile phase depending upon that polarity of drug. So we can select that like the different mobile phase like methanol water, acetonitrile water, then or any other that methanol or buffer. So in that if you select that buffer, there pH, maintain the pH of that buffer, that is very important. Then role of temperature. So temperature of that column is very important to maintain the temperature of that column. Next, flow rate. So you have to maintain the flow rate of that mobile phase. So when you have to optimize that HPLC method to determine that drug in that plasma or serum, so we have to first select that column, select that mobile phase. So according to that you have to select that mobile phase, then maintain the temperature of that column, and then also select the flow rate. Then after that you have to select the method of extraction. So there are three types of extraction method: protein precipitation method, liquid extraction method, and solid phase extraction method. So any out of three method, we can select any one method. So here advantage of extraction method, that is we have to reduce that organic solvent consumption, easier collection of the total analyte fraction, and more efficient separation of interpresence. So here it is very helpful for that selection of that extraction, sample extraction method. So we can extract the sample from that plasma, serum or urine. The next, bioanalytical method validation. So bioanalytical method validation has been used for quantitative measurement of analyte in given biological matrix, and this bioanalytical method which are reliable, reproducible, accurate, precise, which are intended for that use. So bioanalytical method validation, as per USFDA guidelines, there are different validation parameters like specificity, selectivity, sensitivity, accuracy, precision, linearity, recovery, matrix effect and stability. So what is selectivity? So the ability of an analytical method to differentiate and quantify the analyte in the presence of other components in the sample. So selectivity is evaluated by injecting extracted blank plasma. First you have to inject the blank plasma, and after that to inject the extracted LLOQ sample and compare the response of blank plasma and response of that extracted LLOQ sample with the help of that internal standard. And then selectivity can be determined. Then accuracy. Accuracy is closeness of determined value to the true value. So to determine percentage accuracy which is equal to 100 multiplied by found value minus theoretical value divided by theoretical value. So here the mean value should be within plus or minus 15% of theoretical value. Then precision, the closeness of replicate determination of sample by an assay. Here the acceptance criteria for that precision, that is less than 15% of CV, that is coefficient of variation. So for LOQ value, it is deviation acceptable, that is 20% deviation. Then percentage CV, that is precision, can be determined by 100 multiplied by standard deviation divided by mean. So there are two types of precision, that is intraday precision and interday precision. Intraday precision should be carried out within a day at different interval of time. So we have to prepare that three LOQ sample, that is LQC sample, that is lower quality control sample, and higher quality control sample. So here we have to determine that different dilutions of that LQC sample and HQC sample, and analysis can be done with the help of that intraday precision. Analysis should be carried out at different interval of time within a day. Similarly, interday precision should be carried out for today, after 24 hours and after 48 hours. So analysis should be carried out for that LQC, that is low quality control sample, and high quality control sample. Then recovery study. So here the detector response obtained from an amount of that analyte added and extracted from biological matrix compared to the detector response obtained for true concentration of pure authentic standard. So matrix effects. What is meant by matrix effect? So the difference in response between neat sample solution and the post extracted spike sample is called as absolute matrix effect. So here we have to determine that also matrix effect with the help of bioanalytical method. The next, stability study. Here there are three types of stability study: freeze and thaw stability, short term stability, long term stability. So first we'll see that freeze and thaw stability. So stability is determined after 3 freeze and thaw cycles. So here 3 aliquots of low concentration of quality control, that is LQC sample, and 3 aliquots of that high concentration, that is HQC sample, are prepared. Then first these samples are kept in freeze for 24 hours, again thaw at room temperature, that is 1 cycle, Again keep in that freeze for 24 hours, remove and thaw at room temperature. Again keep in freeze for 24 hours and thaw at room temperature. So 3 cycles can be, so like that here cycles can be performed and then stability is determined. Then long-term stability study. So here time in between date of first sample collection and last samples analysis should be carried out. So stability study can be carried out in between that 12 to 15 days. Then stock solution stability, it is evaluated at room temperature for at least 6 hours. The analysis carried out for that sample solution for 6 hours. So here stability study carried out for that LQC sample and HQC sample, and determine stability study for that according to that 3 different methods, that is freeze thaw stability, long term stability and stock term or short term stability study. So bioanalytical method development is very useful for that determination of that drug present in biological fluid or in that biological matrix like serum, plasma or urine. So it is a quantitative method. So here it is determined with the help of HPLC method, LCMS method or LCMS method. And these are here mostly hyphenated techniques are used for determination of drug and also its metabolites present in the biological fluid. So this is very helpful for in industry for that bioanalytical study, bioanalysis study, then viability study, pharmacokinetic study. So here for that we have to find out that bioanalytical method development and we have to develop that bioanalytical method for determination of the drugs present in the biological fluid with the help of that USFDA study. So here we have to apply that USFDA guidelines for the development of bioanalytical method for estimation of drugs present in the biological fluid. Thank you.